In the last video, we took a tour of the machinery of photosynthesis that you find inside of leaves, and that tour ended talking about thylakoids, which are the thin green discs that you find stacked up inside of chloroplasts. And I mentioned that all of the work of actually capturing sunlight, capturing energy from the non-living realm and bringing it into the biosphere is accomplished at the thylakoid membrane. So this phospholipid bilayer, just like any other membrane we've talked about, it's a phospholipid bilayer with proteins floating around inside of it. It's able to do this very, very complicated metabolic task. This first step of photosynthesis is referred to as the light-dependent reaction. So that's what we're going to talk about today. What is happening on these thylakoid membranes, the light-dependent reactions, and that is the first half of photosynthesis. Photosynthesis has two parts, the light-dependent reactions and the light-independent reactions. So today we're just talking about the light-dependent reactions. Now, if you've taken nothing else away from my lectures on biochemistry and how cells work and biomolecules, you've probably taken away that proteins do just about everything inside of cells, so this is not going to be much different. We're going to talk about a series of proteins that interact with each other that are all floating around in this thylakoid membrane. Because they are membrane-bound proteins, that tells you that the components of light harvesting pathways are nonpolar. These are nonpolar proteins that are embedded in the membrane, and the pigments that are going to be harvesting the light energy are also nonpolar pigments as well. So you'll find these components inside of thylakoid membranes, but if we were talking about bacteria, well, they don't have chloroplasts, but they do have a cell membrane. You'd find these same sorts of proteins and pigments on the cell membrane of blue-green algae, cyanobacteria, photosynthetic bacteria out in the wild. Now, what is the ultimate product of the first step of photosynthesis, the light-dependent reactions, the first half? Well, what's going to happen is that water is going to be oxidized. The oxygen in water is going to be oxidized in order to grab some electrons. And the energy that's captured from sunlight is going to be transferred over to ATP, our old friend, the energy currency of the cell, and another high-energy coenzyme called NADPH. This is another nucleotide coenzyme like ATP. This is actually a dinucleotide coenzyme. And I remember the P in NADPH means photosynthesis. This is the one that you see in photosynthesis. Later, we're going to have a coenzyme in cellular respiration that's just NADH. So there's two of them. The one with the P is the photosynthesis one. And you might see that and say, well, I don't know too much about this NADPH stuff, but ATP, hey, that's the energy currency of the cell. Job done. Light-dependent reactions is all I need. I just make ATP from sunlight. That sounds like a pretty good deal right there. Why do we even need to keep going further? But we're only going to use those coenzymes, the ATP and the NADPH, in order to power the process of putting together carbon dioxide molecules to make glucose molecules, and that happens in the second half of photosynthesis, the light-independent reactions. So the purpose of the light-dependent reactions is to capture enough energy from sunlight just to build up our energy reservoir of ATP and NADPH that we can then use it to complete the second half later on. This is also going to be the phase of photosynthesis where we make oxygen as well. That's going to be the first thing that happens. Well, wait a minute. How do plants even do this? I mean, we're animals. We think about, getting biological energy, getting energy from organic sources as a very, you eat your food and your food gives you energy, right? But the plants are getting it straight from sunlight. So how are they converting something like sunlight, like just warms your face and it shines light on things, over to chemical energy? How does that work exactly? How are they converting light into chemicals? Well, they're not doing that. They're not converting it into chemicals. They're using the energy of the light to rearrange how chemicals are bound together and storing it that way, as we talked about in some of the other videos a little bit earlier on in this unit. The way that they're capturing the energy of the sunlight is through the use of something called pigments. And you're probably familiar with the term pigment, right? If you are going to paint something, you need to have a little bit of pigment in that paint, right? That determines the color of the paint. And pigments are used in the same sense here. These are any kind of chemical that you might have that is particularly good at absorbing certain wavelengths of light. Now, if you are a compound that is really, really good at absorbing a few wavelengths of light, that means that other wavelengths of light are going to reflect off of you. And whatever reflects off of you is your color. So if, for example, you are really good at absorbing purple light and blue light, and really good at absorbing red light as well, then you will appear to be a really yellowy green kind of color, because that's the only kind of light that bounces off of you. So pigment molecules, when the light hits them, they absorb some of that energy. Well, how does that work? Well, pigments are made out of atoms, just like any other molecule that we've talked about, and those atoms have electrons inside of them. And the weird thing about light is that sometimes it can be treated as a wave, like we talk about the wavelengths of light, and we can talk about all the different wavelengths being different colors, right? White light is all the different colors of the rainbow combined together. But sometimes we can talk about it as particles as well. If a photon, which is a particle of light, it's a massless particle, hits an electron, the electron can absorb that photon. So that little packet of light, that little unit of light can actually be absorbed altogether. Light doesn't behave in the same way that other particles with mass would maybe behave in this sense. And all the energy in that photon will then be captured by that electron. What happens to that electron? It gets excited. It moves up to a higher energy state. It moves up essentially to a higher orbit, right? To a higher energy shell. And what do we know about bonding, right? It's the valence shell electrons that tend to do the bonding. So if you move up the electron, it actually is going to tend to form bonds more likely. It's going to be more energetic. It's going to be more likely that it's going to move from one molecule to another molecule, right? It's going to be passed around. So when pigment molecules capture light energy, they can pass that light energy from one molecule to another molecule. What's interesting is that in the thylakoid membranes, you have these protein complexes, these big protein complexes. Here's a picture of one on the slide right here. This one's called Photosystem 2, and it's the first step in photosynthesis. In the light-dependent reactions, Photosystem 2 is our first protein. And it's got all of these pigments worked into its structure. It's got chlorophyll A, it's got chlorophyll B, it's got carotenoids, it's got xanthophylls, all of them kind of worked in to this big old protein. And all of these pigments are absorbing different wavelengths of light. You can see here on this chart, chlorophyll A, pretty good at absorbing violet light, pretty good at absorbing reddish-orange light. Chlorophyll B, really good with the kind of blue-violet light, pretty good with the orangey light. Carotenoids, pretty good with the blue light. So you have all these different pigment molecules. None of them are absorbing the green light, so we see plants as being green as a result. It's the only frequency of light that's being bounced off of them. Now, what we have on the slide just below that is a graphical display of some results from an experiment that was conducted in 1883 by a scientist by the name of Theodor Engelmann. In this experiment, he illuminated a filament of photosynthetic algae. So this is a protist that is able to do photosynthesis as well. He passed the light through a prism, though, so he has this rainbow gradient of light all across these different segments of these algae. So you have different wavelengths of light, so the algae can conduct photosynthesis at whatever efficiency they will under red light, under orange light, under yellow light, under green light, under blue light, under purple light. So the whole tank is filled up with this algae, and it's all doing photosynthesis, but they're exposed to different types of light. Then he added an aerobic, non-photosynthetic bacteria. So this is a bacteria that needs oxygen in order to survive and can't do photosynthesis for itself. And he found that the groups of bacteria were largest in the areas that were illuminated by the blue and the red light, showing that photosynthesis is actually most efficient everywhere but the green section of light. So if you were to ask plants what their least favorite color is, they would say green. Green is the color we could do without if we had to pick one. But I digress. So we have photosystem 2 here, this big protein floating around in the thylakoid membrane. And there's all these pigments inside of photosystem 2, and they're capturing all these different wavelengths of light, and they're transferring all of that energy. It's going to their electrons. They're able to transfer that energy from one pigment to another pigment through what's called resonance. We're getting a little bit quantum mechanic-y in photosynthesis and a lot of chemistry and things like that. You tend to get just into the territory of quantum mechanics, and then we always steer away at the last second. So you just need to know that all of the pigments in this photosystem can take all of the energy they've captured, and it can be concentrated to one particular pair of pigments in photosystem 2. The special pair of pigments is called P680 for photosystem 2. And in P680, we're going to concentrate all of that energy to one electron, a high energy electron, which is then going to be passed from photosystem 2 to another protein in the system, to a mobile electron carrier protein to take it to the next step of the light-dependent reactions. This is going to begin what is called an electron transport chain. We're going to keep passing that high-energy electron like we're playing a game of hot potato from one protein to another protein to another protein to another protein to another protein until it finally ends up on that coenzyme I mentioned a little bit ago, NADPH. That's the final electron acceptor here. But here, we're at photosystem 2, and we have something else to deal with at photosystem 2. Because photosystem 2 just took all of its energy, put it in an electron, and then gave that electron away. And as a result, it's down one electron, and it has a pretty potent charge now. It just gave away a negative, so now it's got a positive charge. It needs an electron to make up for that charge. It needs to steal an electron from somewhere else. Where's it going to steal it from? Well, there's all this water hanging around inside of the chloroplast. We have a constant stream of water coming up from the roots. So we're going to take some water molecules. We'll take 2 water molecules, and we are going to rip all of the electrons away from the bonds between the hydrogens and the oxygens. And we're going to be left with an O2 molecule, oxygen gas, 4 hydrogen ions, protons, just floating around in solution. Those will go into the thylakoid space. We'll see what happens with those later on, but those will be important, those protons, positive charges floating around. And we get some electrons freed up, and then those electrons can be used to charge up and then pass on to the next protein. Charge up and then pass along, charge up and then pass along. And then when we run out, we grab another couple of water molecules and do this again. This process of using light to break water molecules is called photolysis. Lysis! Our old friend Lysis is back again. Lysis, to break, and here, photolysis, to break with light. What are we breaking in this scenario? We are breaking water molecules. That is what's being broken here. Now, what happens if you lose hydrogens or electrons? A chemical reaction wherein something loses electrons. Oil rig. Oxidation is losing, reduction is gaining. Okay, oxidation is losing. Oxygen is being oxidized in this reaction. That's not supposed to happen. Oxidation is named after oxygen. Oxidation is what oxygen does to other things. Photosynthesis is so powerful, it oxidizes oxygen. That's amazing. That doesn't happen in basically any other biological process in the entirety of biochemistry. That is incredibly powerful. That is how much power there is in this captured sunlight energy, that we are able to oxidize oxygen and produce O2 gas. Photolysis is responsible for every molecule of oxygen that you have ever breathed in your entire life. It's 20% of our atmosphere, 21% of our atmosphere is O2. You can thank photosynthetic algae, photosynthetic bacteria, photosynthetic plants, all of them using this photolysis process. You can thank photolysis for that oxygen. So know this part for the exam. Know that photolysis is just about the first thing that happens in the light-dependent reactions. Know that it is using the power of sunlight to break water molecules and convert it to O2 in order to replace the electrons that are lost from photosystem 2. And know that this is the step in which O2 is generated in photosynthesis. So, the light-dependent reactions require a constant source of light, of sunlight, and a constant source of water. Everything else is already in the cell for them to assemble, but they need a constant source of sunlight and water. And that's why they're called the light-dependent reactions. The other half of photosynthesis doesn't directly interact with sunlight, but we'll get there in a bit. Now, I know that was a lot of content with photosystem 2, but it is because there was a lot of important things going on there. A lot of the rest of these proteins we won't need to talk about in nearly as much depth. I'm just going to give you a kind of overview of what their functions are. So, photosystem 2. Filled up with different pigment molecules, capturing light energy. They transfer all of that energy through resonance to a special pair of pigment molecules called P680. They put all of that energy into a high-energy electron, which is then transferred to a mobile electron carrier, which we call plastoquinone. There's nothing special to remember about that, except that it is what carries the high-energy electron from photosystem 2 over to cytochrome B6F, it's called, cytochrome B6F. Cytochrome B6F is a pump. It pumps protons, hydrogen ions, into the lumen. So it pumps them from the stroma of the chloroplast into the thylakoid space. So it pumps things into the thylakoid. And that fills up the thylakoid with positive charge as it goes. Now, where does the energy for cytochrome B6F to do that come from? It comes from the high energy electron. So once it gets the electron, it pumps a little bit of these hydrogens into the thylakoid, and then it passes the electron over to another mobile electron carrier protein in this thylakoid membrane called plastocyanin. And then plastocyanin takes that electron over to photosystem 1. Photosystem 1 is very much like photosystem 2. It's full of pigments, it captures light energy, it passes all of that energy to a special pair of pigment molecules. This one's called P700. The difference is that there's no photolysis at photosystem 1. Photolysis only happens at photosystem 2. Because photosystem 1 has a constant stream of new electrons coming from further upstream of this chain here. You can see that it's getting its source of electrons from all of these other proteins carrying them to it. So it doesn't need to capture them from water in order to produce oxygen. So what happens at photosystem 1? Well, basically that electron was used at cytochrome B6F to do a little bit of work, so it's lower in energy now. It needs to be recharged. So photosystem 1 is a recharge station. Now, why is the first photosystem called photosystem 2 and the second photosystem is called photosystem 1? Because we discovered photosystem 1 first and photosystem 2 second, and then we figured out that actually 2 came before 1. And sometimes in science, it would be too hard to convince everyone to change how we've named things. And so we don't. And it is the students who end up suffering as a result for it. And I can apologize on behalf of all biology professors. And the only solace I can give you is that every biologist was just as confused as you were when they were first presented with this information, but we all soldiered forward and memorized it. So we know how bad it is, but that's just the way it is. Okay, so photosystem 1 is a pit stop where that high energy electron gets recharged, and then it gets sent over to another mobile electron carrier called ferredoxin. Ferredoxin carries that, it's actually physically connected to another protein, which is called NADP plus reductase. Ooh, I called it a protein, but A's, that's an enzyme, and enzymes are cleverly named after what they do. It's a reductase. Okay, so it reduces something. What does this enzyme reduce? What does this reductase reduce? It reduces NADP plus. This is NADP plus right here. It's showing it coming from the other side, and it's a coenzyme. It's a nucleotide. It's a dinucleotide coenzyme. And to reduce is to add electrons or hydrogens to something. Well, in this case, we're doing both. We're going to slap this electron that we've just been watching its progress through this electron transport chain on to NADP plus in order to get NADPH, which was the coenzyme that I told you towards the beginning of the video was one of the ultimate products of the light-dependent reactions. So you know how I said that for ATP is like the charged up battery and ADP is like a half-used battery? NADPH is the high energy form. It's the ready-to-use form of that coenzyme. And NADP plus is the less charged. It's the uncharged version of that coenzyme. This is just another energy carrier like ATP is, but a slightly different form. ATP carries its energy in its phosphate groups. NADPH carries it in the form of a high energy electron, which is attached to this hydrogen at the end. That's what the H on the end is a hydrogen with a high energy electron on it. Remember in the redox video when I told you that sometimes we follow electrons and sometimes we follow hydrogens? This is the kind of thing I was talking about, where throughout this entire electron transport chain, remember this is all in the thylakoid membrane. These are all just proteins bumping into each other. We've been following this electron going from photosystem 2 to PQ to cytochrome B6F to PC to photosystem 1 to FD to NADP plus reductase over to its final resting location, NADPH. But then when we finally get the electron on NADPH, we don't look at it as an electron, we just see that there's an H on the end, and we know that that is where our electron is. Now, oil rig, oxidation is losing, reduction is gaining. It's gained an electron, and it's gained a hydrogen, so this NADPH is the reduced form of this coenzyme, and that means that this can now go forth and reduce other molecules. It can kind of play tag. It can go place that hydrogen on something else now. Now that is the end of the electron transport chain for the light-dependent reactions, and we've made NADPH, and we also made oxygen along the way as well. But we still haven't made ATP, but I'm going to talk about that in the next video, how we get ATP out of this process. To finish this video, I wanted to try to summarize the electron transport chain in a slightly different way, because I know it seems like we just suddenly entered a brand new class, right? Like it suddenly the amount of biochemistry language just escalated by an order of magnitude. And I wanted to show it more visually. So I sat down and I tried to animate the electron transport chain in the hopes that that would help it make a little bit more sense. So here you can see that there are 4 large proteins. There are 4 important stops here. Photosystem 2, where photolysis happens. Cytochrome B6F, where hydrogen ions are going to be pumped into the thylakoid. Photosystem 1, where the high energy electron that's being passed along is going to get a recharge, and then NADP reductase, where we are going to reduce that coenzyme into NADPH, which we will need for a later step. Now here I'm going to hit play, and you can see photolysis beginning, right? We have 2 water molecules coming in, and we have the hydrogen ions forming. So we have these positive hydrogens. Photosystem 2 is taking the energy from these photons that are coming in and shuttling it into these high energy electrons. And then what do we have? These mobile electron carriers, PQ and PC, they are carrying them from one location to another. So at cytochrome B6F, we can see that there are hydrogen ions being pumped into the thylakoid. And then over here at photosystem 1, there's no photolysis happening, but we're getting more photons, more light energy being captured. And then ferredoxin carries the electron over to NADP reductase, and now we have some NADP coming in and being reduced to NADPH. So that's what this is. Here you can see that the electrons are coming in on the water, they're getting charged up for the first time at photosystem 2, and they're just being passed down the line by these different proteins which are embedded in the thylakoid membrane. And there's a couple things happening. One, we're eventually putting that high energy electron on NADPH, but two, we're pumping hydrogen ions into the thylakoid, which is creating a whole bunch of positive charge down here. You can see all these positive charges starting to build up. Now, how we deal with those positive charges is going to be related to how we get ATP out of this process. So that is where I will pick up in the next video.